Okay, so welcome back to this next video in which we are discussing cancer development. Okay, right, so we've discussed that in cancer development, what's going to happen is you start off with a single cell which acquires some heritable change that then allows it to overdivide. Okay, so it then overgrows and overdivides and produces a whole clone of cells which are all capable of overdividing. Okay. Now, in that clone of cells that have overdivided, which is now called a benign tumour, okay, within that clone, one of these cells is now going to get another heritable change, okay, and the heritable change that then comes next is going to take you closer to becoming cancer, okay. So the idea that underpins our theory of cancer development is that it is this gradual process of accumulating these inheritable changes. And note that I do not say specifically whether it's a mutation or an epigenetic change. The way of combining those together is these heritable changes. Okay, so you'll gradually have to progress along this process. Now, one of the heritable changes that you acquire very early on is a change that results in problems in DNA repair mechanisms, okay, or potentially chromosomal segregation during mitosis, okay, which then leads to genetic instability. Now, in the model that I'm presenting to you, we're putting this as the second one. It's not necessarily the second one. It's one of the ones that you're going to acquire in this process. So it will occur at some point on the way to cancer. It's a very important one, and it's generally going to occur early. The reason being that getting this genetic instability is extremely useful for then going through more stages after this, because if you've got a population of cells that are undergoing mutations at a rapider rate, that means that the rapidity that further mutations are going to occur, that are going to take you further along the way to cancer, is going to increase. Okay, so generally this genetic instability is acquired early on, so we'll put it as the second one. Okay, what we are now going to talk about is the next mutations that occur, and now we're not going to do these in any specific order. These are the mutations that by the time you've got to cancer, you will have, okay? So, you can imagine, if you like, that the one that I'm about to say is the third mutation. So you can imagine that one of these cells here, okay, in this purple clone of cells, which remember are the cells which have both the heritable change that allows them to overdivide and also the heritable change that allows them genetic instability. Okay, you can imagine that one of these cells is now going to get a further mutation that's going to be advantageous. Okay, and the next mutation that I'm going to talk about is changes in metabolism. Okay, and this is known as the Warburg Effect. Okay, so to understand the Warburg Effect, which is named, by the way, after Otto Warburg, who was the first person to describe this effect, okay, to understand this effect, you need to understand that in these tumours, oxygen levels are going to be very, very low. Okay, you've got a lot of cells that are very, very hungry, which are all trying to metabolize, okay, and they haven't got a very good blood supply at the moment, okay, because this is a new growth, okay, so it hasn't got a very good vascularization yet. Okay, so you're going to have very low levels of oxygen in there. Surviving in low oxygen levels is not easy. Okay, so if you gained a mutation that then allowed you to survive well in low oxygen conditions, that would allow you to survive better than your neighbours, and therefore you would most likely grow and divide more than your neighbours. Okay, this is one of the mutations that is found, or heritable changes I should say, rather than mutations, because it could be an epigenetic change as well. This is one of the heritable changes that is found in the eventual cancer cell, which is right at the end of this consecutive process of acquiring these heritable changes. Okay, so let me now describe what the Warburg Effect is then. Okay, so firstly, let me tell you about normal metabolism within cells. So in normal metabolism within cells, you take up glucose, okay? The glucose then goes through a series of reactions which are collectively known as glycolysis, Okay, so glycolysis is not a single chemical reaction. There are a huge number of reactions within glycolysis. But the overall product of glycolysis is that you get pyruvate molecules. You get 2 pyruvate molecules at the end of it. Okay, then these pyruvate molecules go through the Krebs cycle, and then they go finally onto the electron transport chain. So I will just put oxidative phosphorylation. And this oxidative phosphorylation process involves oxygen. If you have low levels of oxygen, this process will stop, basically. So this requires O2 to be present, basically. Okay, so if the electron transport chain, well, specifically the portion of the electron transport chain that requires oxygen is right at the end. It's complex 4 that requires oxygen. Okay, but if you don't have oxygen, complex 4 won't work. Okay, and if complex 4 doesn't work, then all of the others further along will back up, basically, and then the Krebs cycle will stop working. Okay, because if the electron transport chain isn't working, then there's no point doing the Krebs cycle, because the Krebs cycle doesn't actually produce ATP, it only produces reduced NAD. Okay, and to get energy out of reduced NAD, you have to have the electron transport chain. So there's no point in going any further after glycolysis if you haven't got oxygen present, basically. Okay, glycolysis itself does produce some ATP, so the biological energy currency is ATP. So glycolysis is capable of producing a little bit of ATP from ADP. Okay, so this first bit is producing some ATP, but to get the most ATP out of the glucose, you need this portion here, and that requires the presence of oxygen. Okay, so this is what normal cells are doing, and this is producing a huge amount of ATP. Much more ATP will be produced through oxidative phosphorylation than through glycolysis. Okay, right. Now, the cancer cells are going to be in low oxygen conditions, so we can say goodbye to the oxidative phosphorylation, basically, or at least it's going to be at very low levels. Okay, so what's actually going to be true in cancer cells is that they're going to acquire mutations in the metabolic machinery that then allow them to deal with this extremely low oxygen concentration. Now, the first thing that changes is that their uptake of glucose is actually hugely greater than a normal cell. So cancer cells actually take up 100 times the amount of glucose of normal cells. So these things are pulling glucose from the extracellular fluid at a rate 100 times that of a normal human cell. Okay, this is the origin of why cancer causes cachexia. Okay, so cachexia is the fancy name for the fact that when people have cancer, they appear to sort of waste away. They get very, very thin, they get very, very pale, they look extremely ill. The name for that is cachexia, and we would say that the person looks cachexic. Okay, the reason that the cancer is causing that, everyone knows generally. They know it's because the cancer is using the nutrients of the body, basically. But you might think, but the tumour is tiny compared to the rest of the body. How can this little tumour make such a difference on the body's nutrients levels? Well, this is the answer. The cancer cells are extremely inefficient at using glucose. Okay, they get hardly any energy out of it at all, okay, because they don't put much of it through this oxidative phosphorylation process. Hence, they have to take up huge amounts of glucose out of the blood, okay, and this is why having cancer makes you cachexic in this way, because of the cancer cells' hugely disproportionate uptake of glucose. Okay, right. So let's now describe what happens within the Warburg effect, because we're going to adapt our entire metabolic pathways within these cancer cells so that they can work in this low oxygen concentration and process this large amount of glucose, basically, okay, to yield an amount of ATP that they can actually live off. Okay, so basically in cancer cells, what is instead happening is that you are taking up this hugely disproportionate amount of glucose. Okay, it is then going through the pathways of glycolysis. Okay, and the glycolytic pathways produce many intermediates. Okay, so glycolysis is a multi-staged process. There are many intermediates in the middle of the glycolytic pathway. Some of these intermediates are used by the cancer cells to produce other molecules that are important in the cells. So for instance, amino acids, which are the molecules out of which proteins are built, nucleic acids, which are the molecules out of which DNA and RNA are built, okay, and also lipid molecules, which are the molecules out of which membranes are built. Okay, so we're going to use several of the components in the glycolytic pathway to make other molecules that the cells are going to need to divide. So approximately 10% of the glucose that we are uptaking is going to go into the production of these other molecules here. So 10% is going to go this way. Okay, right. The other 90% is going to go off to pyruvate. Okay, however, the pyruvate doesn't generally go to oxidative phosphorylation, okay, because of the low oxygen levels within the tissue. Okay, so only about 5% of the pyruvate is now actually going to go through oxidative phosphorylation. I might just use the oxidative phosphorylation I've got here. Okay, so 5% of it's going to go to oxidative phosphorylation. And I mean 5% of the original glucose rather than 5% of the pyruvate. So the numbers here are eventually going to add up to 100 basically. I don't mean 5% of the 90%, I mean 5% of the original glucose is actually going to go down oxidative phosphorylation and produce ATP that way. Okay, so this is much lower. Okay, and the other 85% of the glucose, which takes these three numbers here up to 100%, this is going to go down the anaerobic respiration pathway, and it's going to be reduced to lactate, basically, lactic acid, the conjugate base of lactic acid. Okay, so this does not produce energy. This conversion from pyruvate to lactate, it does not produce ATP. All it does is recycle a molecule known as NAD, basically, so that you can continue the glycolysis pathway here. So basically, in glycolysis, you do produce some ATP, but you also produce reduced NAD, okay, which is often written like so. NADH.H+. Okay, so NADH contains NAD with a hydrogen atom and then one electron. Okay, so let me just describe this in a little bit more detail. So basically, NAD is an electron carrier. Okay, it's a molecule that has a positive charge. Okay, what can happen is this molecule can accept what is known as a hydride anion. Okay, which is a hydrogen atom with a negative charge. So this is called a hydride anion. Okay, and a hydride anion is quite rare. It consists of a proton with two electrons surrounding it. So it's a hydrogen atom that has gained an electron. You are probably more used to seeing hydrogen cations, okay, rather than hydrogen anions in this way. Okay, hydrogen cations are where the hydrogen atom has lost its only electron. Hydride anions are where the hydrogen atom has gained an electron. Okay, so what you can do is attach one of these molecules onto NAD, okay, and this neutralizes the positive charge of NAD, and it produces what's known as NADH. Now, in reality, when we look at these biochemical reactions, what is happening is these biochemical reactions are giving up two hydrogen atoms. Okay, I shouldn't put it like this. The biochemical reactions generally give up two hydrogen atoms, i.e. two protons with two electrons. So you have a hydride anion in there, but you also have the other proton. Okay, so what do I mean by that? Well, if you've got two hydrogen atoms, you've got two protons there, and also two electrons. Okay, that is the equivalent of one hydride anion, which is a proton with two electrons, and also one extra proton. Okay, so often people write NADH.H+ to remind us that we've really got an NADH molecule, a reduced NAD molecule, with this proton floating around as well. Okay, so this reaction gives these two hydrogen atoms, the hydride anion from the NAD, reduced NAD molecule, and the hydrogen cation that's also come from this reaction to the pyruvate molecule to convert it into lactate. And all that does is it regenerates the original NAD plus molecules back again. Okay, so that's the only use of that reaction. It means that we now have these molecules back again so we can do more glycolysis. Okay, so this is the anaerobic respiration reaction. This is what is happening in your skeletal muscle when oxygen levels go very low. Okay, and it results in the production of lactate. Okay, so cancer cells have hugely upregulated anaerobic respiration, basically, and this is incredibly inefficient at getting ATP out of glucose, hence why cancer cells need this extraordinarily high uptake of glucose. So, this overall change in the metabolism of cancer cells does require heritable changes in the cancer cell. Okay, it's not just a physiological response. You might think, well, surely if I take any cell and put it in extremely low oxygen concentrations, won't it do this? Won't it change its metabolism to cope with that? And the answer is no, not really. Basically, these cancer cells, even if you take them out of the really low oxygen concentration environment and put them in a perfectly normal oxygen environment, they continue this process, basically. They continue this process, this really bizarre metabolism. So this metabolism is not just a response of the cell to the low oxygen concentration. It's not just a response that can be turned off. It is a permanent change in the cell's metabolism that persists even if it's put in a high oxygen environment. Okay, right. So it does require heritable changes. So at some point, one of these in this process towards cancer, the cell is going, one of the cells in your current clone is going to get a heritable change that leads to this shift in metabolism. Potentially it's more than just one heritable change, maybe you need multiple, okay? At some point though, you're going to get this shift in metabolism towards this Warburg metabolism, okay? And that's going to better able these cells to survive in this environment, okay? And therefore they will over-proliferate because they do better than their surrounders, their neighbours. Okay, so if we imagine that this is the third mutation in our tumour, okay, so let's say here is our tumour, okay, then we've got our purple population of genetically unstable cells, and let's say we've now got a huge new population of these cells that have undergone the Warburg effect, basically. So here is our tumour, let's say, and here is the rest of the duct going off there. So, let's say that these are the cells, the purple cells, which remember are the cells which are genetically unstable and which are over-proliferating. You will still have some orange cells, which are the ones which just have the first initial mutation of over-proliferating, but gradually these cells are going to diminish because they're going to be outcompeted by the more recent mutations. Okay, so let's say that this new population here, which I'm now going to colour in turquoise, these are the cells which have just acquired this heritable change that means that they can have this adapted metabolism which will help them survive in the low oxygen conditions of this tumour. Okay, then we've still got some of these purple cells here, which are the ones with just the genetically unstable they have genetic instability and they're over-dividing. And then we have the orange ones, which just over over-division. Okay, right, so we're going further along, basically, now. Okay, right. So, because the Warburg metabolism helps you survive in low oxygen conditions, these turquoise cells are now going to do better than their neighbours, and they're now going to grow and grow and grow, producing a whole clone of cells which have all three of these mutations now. They are genetically unstable, they are over-dividing anyway, and they have brilliant metabolism, okay? And this population is going to outcompete all of the neighbours, so it's going to start destroying these older cancer cells. I mean, some of them will survive, but they're going to start being outcompeted by these newer ones. Okay, right. And I want to stress again that the order in which I'm showing these mutations is not necessarily set, okay? It's not necessary that these mutations occur in exactly this order. This is just to give you, so that I can draw pictures at all, I need an order to it. Okay, but this could have occurred much later, basically, but this is one of the mutations that by the time you actually get to cancer cells, they will have acquired. Okay, right, so let's now talk through the next... Oh, actually, before we go on to the next permanent change, I just want to talk about a clinical correlate of the Warburg effect, okay, because this is quite interesting. So, these cancer cells, a way of telling them apart then from normal cells is that they have this hugely increased uptake of glucose. So they take up glucose 100 times faster than normal human cells. Okay, we can take advantage of this fact, okay, to find cancer. Okay, we can use this to diagnose cancer. How? Well, basically, we use a molecule known as fluorodeoxyglucose. Okay, and now for short, fluorodeoxyglucose is often abbreviated as FDG. Now, this is not just a normal fluorine that you have in fluorodeoxyglucose. Okay, you put a very strange fluorine atom in fluorodeoxyglucose. You put in a radioactive isotope of fluorine. 18 fluorine is the one that you put in. And 18 fluorine is special because it can emit a form of radiation known as beta-positive particles. Okay, so we talked about, when we were discussing ionizing radiation, we talked about beta-negative radiation, which is a more common form of radiation. Okay, and beta-negative radiation was electrons. Beta-positive radiation is small particles known as positrons. Okay, now these are the antimatter particle of electrons. Okay, so here is a positron. Now what's significant about that is that it has firstly the opposite charge to the electron. It has a positive charge rather than a negative charge. And also, more importantly, when a positron collides with an electron, okay, so let's say we've got an electron here, and electrons are all over the place. You know, all atoms have electrons in. Positrons are much, much rarer, and that's one of the great mysteries of physics, why antimatter particles are so much rarer. Okay, but when positrons and electrons meet, what happens is they annihilate each other. They go. Once they bang into each other, that's it. And when they annihilate each other, they give off very high frequency electromagnetic radiation. Okay, and these are gamma rays that they give off. Okay, so this is the great idea. We put in this fluorodeoxyglucose, which has this 18 fluorine in it, okay, and we put this into the patient's blood. Okay, now this might seem like a crazy thing to do. This stuff sounds dangerous, but yes, it is radioactive, but it's worth it for the diagnosis of the cancer. Okay, what then starts happening is that the cells of the cancer, if they are present, so if you do have cancer, what will happen is the cells of the cancer, so let's just draw the cancer tumour here, so here are the cells of the cancer, they will start uptaking the fluorodeoxyglucose at rates 100 times greater than how peripheral cells will take it up. Okay, so cancer cells are going to take up fluorodeoxyglucose, this radioactively labelled analogous molecule to glucose, 100 times faster than normal cells. So you're going to get loads of this stuff in the cancer cells. The idea then is that it will give off the radioactive particles, so it will give off the beta-positive emissions. So you'll get loads of positrons being emitted here. They will bang into electrons, they will annihilate, and I should have just put that this process is known as annihilation, or annihilation, okay? And then you're going to get these gamma rays being given off. So what you then can do is you can detect the gamma rays, because gamma rays are extremely inert. These things will pass through meters and meters of lead, basically. Okay, so they're certainly going to get through patients' bodies and out to detection machinery. So what you'll have is you'll have your patient here, okay, who you've given the fluorodeoxyglucose to, and now what you will try and do is detect gamma rays coming from the patient, and you'll look at which portions of the body these gamma rays are coming from, and that can help you to detect where cancer is. Okay, and the idea is that cancer, you will have a huge, well, where the cancer cells are, you'll be getting a large amount of these gamma rays coming from those cells because they have taken 100 times more of the fluorodeoxyglucose than normal cells. So you would expect their gamma radiation to be 100 times more. So you will see it as a very bright patch, basically, if the cancer is present. Okay, so this is actually one of the key things that we use to diagnose someone with cancer. Okay, so this has a special name. It is known as a positron emission tomography scan. Okay, because of how much positrons are involved in this. So this is known as positron emission tomography. Okay. And for short, positron emission tomography is abbreviated to PET. So when people talk about going for a PET scan, what it means is that they are having this radioactively labelled form of glucose inserted into them, which is then releasing positrons, which are meeting with electrons, annihilating, releasing the gamma radiation, which is then being detected by a machine. And that allows us to decide whether someone does have cancer or not. Okay, so that's a nice clinical correlate of the Warburg effect. Okay, so that's one of these mutations that you are going to, or I should say heritable changes. I'm sorry, this is a running theme that I'm forgetting to say heritable changes over mutations. Okay, remember that they can either be mutations, i.e. changes in the genetics, or they can be these changes in the epigenetics. I should say heritable changes. This is one of the heritable changes that you need to have acquired in the process towards cancer. Whether it's the third one, that's, it's not going to be the third one in all cancers, okay, but for the basis of having a model, we can certainly imagine that it's the third one. It makes it easier to understand, certainly. Okay, so we'll go along and look at another heritable change that you need to get to as you develop towards finally being a cancer cell in the next video.